Hi everyone, can you hear me? So it's a long title, so I will get to that slowly. And spoiler alert, this work has been done in yeast. So we move from butterfly to yeast into single cell organisms. So what do I mean by cryptic variation? So there are various functions that are performed by different proteins in an organism. It could be binding to DNAs, it could be forming protein complexes, or binding different molecules. But there are certain processes that are conserved across organisms, and let's say this particular protein sequence performs a specific function in the human. There are other protein sequences in orthologous proteins which perform the same function in different organisms. Now what we see is that there are certain positions which have a specific amino acid present while the others have a lot of variation. And this variation which doesn't affect the function in the organism but is present is what we define as cryptic variation. And given that we saw that there are different kinds of sequences, we have a different sequence in yeast and human. And if we try to create a particular mutation at the second position over here, there's no effect on the function in the yeast, and the yeast is fine. But if we try to do the same mutation in humans, the human might die. And this phenomenon where the same mutation has different effects in different protein sequences is what we call as molecular epistasis. Now, what do I mean by redundant duplicate genes? So imagine in an ancient organism, there is a specific gene which performs these three functions. And for following a duplication event, you have two copies of the gene which perform the exact same function. And over time, they accumulate various mutations, which changes their sequence, but it doesn't affect their function. And it has been estimated that approximately 30% of genes in yeast have high level of functional redundancy. And over longer time scales, like hundreds of millions of years, the two major outcomes of gene duplication are essentially non-functionalization, that is one of the copies loses all the functions and degenerates, while the single copy maintains all the functions. Or there's an example of subfunctionalization where one of the copies maintains some functions while the other maintains the other, and overall the two copies maintain the ancestral function. So the question that I want to answer here is, does epistasis influence the evolution of proteins after gene duplication? For instance, like if you create the same mutation in these two gene duplicates, do you have the same effect or different effects? Like in this example, if you create the mutation in this gene, it loses all function. But if you do the same mutation in the other gene, there is no change in function. And for the purposes of this talk, the way I define functions is through protein-protein interactions, that is, different protein complexes that are necessary for carrying out certain functions in yeast. And the model system I use is something called as an SS3 domain. Now, SS3 domains are short amino acid sequences of around 60 amino acids long, and they are well folded. And they bind to various intrinsically disordered regions in other proteins, and that is how they take part in signaling pathways. And the way they do that is a particular SS3 domain would recognize a specific amino acid sequence on the disordered regions, and that's how they bind to them. And these are two examples of those. And these large letters basically mean the positions that are quite important for binding. And the proteins I'm interested in working with are duplicated myosin proteins. Now, myosins, as you might have heard, are muscular proteins, and yeast don't have muscles. In this case, these myosins are involved in various processes which are necessary for providing shape to the cell. And these two SS3 domains have almost 20% coding sequence divergence, which is approximately 12 amino acids out of 60, which are different. And the interesting part is that they bind to the same specific amino acid motif, which is that the function is the same. And we identified three such proteins in our experiments, which have the specific binding motif. And the way we test the effect of mutations is through an experiment called as deep mutational scanning, where we create all possible single amino acid mutations across the SS3 domain in both these duplicated genes, and then we test their effects on binding. So, what do the effect of these mutations look like? So in this slide, I'll show you the color bar, where 0 means that the protein interaction is totally lost, and anything in blue means the protein interaction is maintained at wild type levels. And the x-axis over here represents the various positions in the SS3 domain, and the y-axis basically means what kind of mutation we create in each position. And this is an example of something we call as a functional landscape, where you see that there are certain positions where creating a mutation kills the interaction totally, where other positions it has no effects, and some positions there is a spectrum of effects. So the next question is, since we test various interaction partners, is the functional effect of these mutations same across them. So what we see for many positions is that the effects are the same, whether we test it for one interaction or the other. But there are several other positions where creating the same mutation has different effect or different protein-protein interactions. So for instance, if you see over here, a lot of these aromatic amino acid mutations, they kill the interaction for this interaction over here with the OS2 protein, but for the other two, the interaction is maintained. And you see various different patterns overall. So we see that the functional effect can change based on which interaction we are trying to study. So this means that any further analysis we do, we have to take this into account. So the next thing we wanted to see was between the two duplicated genes, is there an effect in this functional effects? And what you see over here is we have the myo3 gene on the x-axis and myo5 gene on the y-axis. And you see that there is like a shift towards the myo5 gene in terms of functional effects. That is, if you create the same mutation in the myo3 gene, it tends to kill function much more frequently compared to the myo5 gene. So what is going on over here? One of the core hypotheses that we looked at first was that a lot of duplicates undergo regulatory divergence immediately after duplication, that is, the expression changes. So that's what we tested first. And what we saw was that we basically took GFP genes and we deleted the actual gene from instead of the promoter and inserted the GFP back at that location. And then we tested by cytometry what the expression levels were. And we saw that the myo5 gene has a much higher expression compared to the myo3 gene. So does this contribute to the effect that we saw? So in order to do that, since we tested everything in, let's say, the myo3 SS3 domain present at the myo3 protein and myo5 SS3 domain at the myo5 protein, we decided to swap them around. We took this SS3 domain and put it here, and took this and put it back here. So now we have the swap domains, and we can compare them across the same proteins or at the same SS3 domain. So what we saw is that when we see the effect of the myo3 SS3 mutations at two different proteins, we see that the same pattern of bias towards the myo5 protein remains. But when we test the two different domains at the same genetic loci, this is at the myo3 protein, we see that that bias disappears and these functional effects are equivalently present in both directions. So what we see here is that the regulatory divergence that is present between these two duplicates contributes to a buffering of the loss of interaction for the higher expressed duplicate compared to the lower expressed one. And when we take the regulatory divergence out, there are some mutations which are favorable in one compared to the other. The question is, how do these effects of mutations vary without regulatory divergence? And the examples I'll show you here are mutations that if you create them in the myo5 SS3 domain, the function is present, but when you do it in myo3, the function is killed. And the reverse case over here. And there were many mutations where we saw that creating the mutation in myo5 SS3 domain didn't affect the function too much. But when we did it in the myo3 SS3 domain, the function was killed. And the reverse case was also present. So this brings me on to the next part is, since we saw that the effect of mutations varied across interaction partners for certain positions, is that thing also present when we look at duplicates? So what we see in this example where we did a valine to leucine mutation is that the effect of myo5 SS3 domain is basically that it maintains the function, and when we do it in the myo3 SS3 domain, the function is killed. And this is something that this effect is present across all the interactions that we tested. But for another example where we have, let's say, proline to a valine mutation at position number 19, we see that it doesn't change much for the BBC1 interaction or the PAN1 interaction, but there's a clear difference for the OS2 interaction. So what we see here is that there are certain cases where the interaction is killed for all interaction partners, but in some cases it's specifically killed for one. And going back to the major consequences, this would be an example where if you create the mutation in myo3 SS3 domain, it leads to non-functionalization of the duplicate, but doing the same mutation in the myo5 SS3 domain has no effect. And this would be an example of subfunctionalization, where creating the mutation in myo5 SS3 domain doesn't have much of an effect, but doing it in the myo3 SS3 domain causes it to lose some of its ancestral functions. So we wanted to see what is the proportion of these mutations present across all the mutations we tested. And before I go on, is that around 30% of mutations kill all the interactions irrespective of the duplicate we introduce in. And when we do the same mutation in either of these genes, the proportion that leads to subfunctionalization is around 2% for the myo5 SS3 domain and around 4% for the myo3 SS3 domain. And for the non-functionalization case, we have around 4% which lead to non-functionalization for the myo5 SS3 domain and around 4 to 5% for the MAO3 assisted domain. So we see that almost 15% of mutations have very different effects when introduced in either domain. So to summarize this, we see that mutations impair binding in an interaction-specific manner at various different positions. And this is interesting because the binding interface is same for all those peptides that we tested. And secondly, regulatory divergence buffers the loss of interaction for the higher expressed duplicate. And thirdly, the epistasis that we see due to the coding variation present between the duplicates dictates the fate of these duplicated genes for a lot of these positions. So what does this mean in general for any kind of protein evolution traits that we try to study? Is that any protein, any loci that we see which has various functions, during any kind of environmental change or something, there is a coding mutation which causes loss of function to these ones. But that doesn't mean that the protein will get removed due to purifying selection or something. There could be a case of a regulatory mutation which increases the expression level and tries to bring back certain functions, or a coding mutation which interacts with another varying residue and is able to bring back that function together. So the point is that any kind of robustness that we see towards mutations for a specific interaction may not apply to others, but that can be recovered by multiple mechanisms, either through changes in the regulatory sequence or in the coding sequence. So I'd like to thank our SS3 team in our lab and all of you for listening, and Rebecca and Paul for inviting me here, and our funding agencies. And for more interesting results, you could take a look at our preprint or come to see me at my poster J17 tomorrow. Thank you, everyone.
So I think we have time for a couple of questions. There's one question there.
Thank you. That was very interesting, and you churned so many different dials in your experiments that I'm still trying to figure out exactly what you changed. So I have two questions. One of them is, all of your mutations are synonymous or non-synonymous?
We did both synonymous and non-synonymous mutations, and synonymous mutations don't have any effect. It's like a very narrow distribution at the wild type levels. Mm-hmm. And non-synonymous mutations has a bimodal distribution at synonymous and non-synonymous levels.
And my second question is, you saw that MYO3 would compensate.
MYO5.
MYO5's upregulated?
Yeah, upregulated, yeah.
Right. So but MYO3 will compensate when MYO5 isn't, when MYO5 is disrupted. Did I get that backwards? The one where you've got the dots along the main diagonal.
So this is the one?
The one on the right.
Yes.
Yes,
so this
okay.
is, we take the MYO3 locus, and we introduce the two domains. And we also do it in the MYO5 locus, and the effect is the same.
Okay. My question is, did you look to see if expression changed?
Expression after doing this?
Yeah.
So we couldn't, like, after deleting the gene, we cannot test that, but we have some data where we add the GFP after the protein, and that doesn't change too much.
Yeah.
So the effect majorly is
I
coming
meant
from
the expression of the compensating gene.
compensating gene.
Because you're saying there's a big difference in expression. So
So you're saying after we put the domain back in MYO3, do we see an effect in the MYO5 expression level?
yeah.
Yeah, that we're not able to do because doing CRISPR at that point is like it kills the cell because these are essential genes. So too many.
So, okay, so there
manipulations.
could be a regulatory perturbation, but
Yes.
it's not something you can measure.
Yeah, it's because it kills the cell. So
Thank you.
Okay, thank you very much. Is there any other question?
Theoretical one. So it seems like, would you say that if a protein has a higher sort of number of binding partners, does this increase the likelihood that you would get subtransmolization?
So what we observed is that, so this is one example that I showed, like this is the model system I work with. We work with other SST domains. So in this case, we see subfunctionation because the nature of the binding pockets is slightly different, because you see the slide where I showed you that the effects vary across interaction partners. So unless that is present, the effect of subfunctionation is very hard to come by, because there is no possibility, like if you kill one interaction, it's likely to kill others. But this is one example where you are able to subfunctionalize the genes. But as you saw, like the percentages are not that high. Like almost 6% of mutations are there where you can have subfunctionation effects. But non-functionalized is much higher, it's like 9%.
So thank you all for coming to my talk, and thank you for the organizers for making this symposium happen. I think a lot of the talks so far have been quite complimentary, so hopefully you find that to be the case with this talk as well. So today I'll be presenting a model of plasticity that examines the effect of sigmoidal reaction norms on evolutionary dynamics. And this work was done in conjunction with my advisor, Bob Holt, and Sam Shiner, who was the one who came up with the idea for this project and is also present in the audience today. So the motivation for this project was to examine how genetic assimilation is affected by developmental constraints to plasticity. And I'm sure people are familiar with this concept, but just to give a quick conceptual sketch, consider a population that is subject to a change in environment that leads to a change in the optimum phenotype. And the resulting phenotypic change in the population will be due to both a plastic response to the environment as well as a non-plastic component of the trait. And initially, this change will be due to plasticity, but afterwards, the initial plasticity will be replaced by the non-plastic component of the phenotype. So genetic assimilation in nature can be difficult to identify, but it has been done in a few cases. I'll just mention the instance of the North American spadefoot toad. So tadpoles in this genus can develop either into an omnivore ecomorph, seen on the left here, if they eat detritus during development, or a carnivore ecomorph if they eat shrimp during development. And in ancestral populations that occur alone, roughly 20% develop into the carnivore morph, and in derived populations that co-occur with a congener, pretty much all of them develop into the carnivore ecomorphs. So the derived populations hatch out as carnivores even when they are exposed to the detritus dietary cues. And we show this with the jaw muscle width. So the derived populations, even when they eat detritus, their jaw muscle width is comparable to that of the carnivore morphs in the ancestral populations. So the populations with the canalized traits express their traits closer to the optimal value and with less performance costs than populations with the induced plastic traits. And the difference in the means for the derived populations between the detritus and shrimp dietary cue is much smaller compared to the ancestral population. And this evolutionary shift towards reduced plasticity was observed in roughly 70 generations. So previous models of genetic assimilation include both quantitative genetic models by Landy and Shevin, as well as individual-based models by Shiner et al. And these all assumed linear reaction norms and essentially no developmental limits to, which essentially means no developmental limits to plasticity. So in these models, genetic assimilation took between 10,000 to 100,000 generations, which is several orders of magnitude larger than what has been observed empirically. So this work is very much building off of the earlier Shiner et al. Models, but keeping in line with the topic of today's symposium, we examined the effect of a nonlinear reaction norm on evolutionary rescue, adaptive plasticity, and of course, genetic assimilation. So previous models use linear reaction norms as seen in this example from impatiens. So plants that grow in a low light environment have a shorter height, and plants that grow in a high light environment are taller. However, in our model, we use a sigmoidal reaction norm similar to what has been observed in the horn beetle. So males below a certain body size remain hornless, and males that exceed a certain body size develop a pair of long curved horns on their heads. So our model is an individual-based model of a population of hermaphroditic and diploid individuals. We simulated 500 discrete and non-overlapping generations, and each individual has a single quantitative continuous trait that's determined by the allelic values at 10 loci, 5 for genes that control the plastic response to the environment, and 5 that determine the non-plastic component of the trait. So in any given generation, individuals are born and undergo development in a particular environment. Plasticity and the environment affect the trait value. The survival of the individuals to adulthood is then determined by the distance of their trait from the optimum. Random mating occurs at the end of the generation. All adults die, and juveniles undergo the same process at the next generation. So initially, populations are adapted to an environment and do not express any plastic response. So in the top right, you should see a reaction norm that's flat. It's a fixed phenotype regardless of the environment. Then they're subject to an abrupt environmental change, which changes the optimum phenotype. And in our model, the optimum phenotype is just equal to the environmental value, and we show the width of the selection function just for visual purposes. And in the next slide, I'll show what happens in response to this environmental change. So the reaction norm has three components. There's a non-plastic component, which is essentially the sum of the non-plastic alleles, which sets the height of the sigmoid on the trait axis. There's a plastic component, which is a function of the development environment, the sum of the plastic alleles, which sets the steepness of the sigmoid, and the developmental limit parameter D here, which determines the maximum range of plastic trait variation. And finally, there's a random component that's unrelated to the genotype or the environment. So when they're subjected to an environmental change, the populations will evolve to increase plasticity expression, so it'll go from that flat reaction norm to the sigmoidal curves we see here. However, because the range of plastic trait variation is limited by this parameter D, this may potentially prevent the ability for plasticity to achieve the new trait optimum. So in such cases, the evolution of a non-plastic component is also necessary in order to achieve the new trait optimum. And if genetic assimilation occurs, then essentially plasticity is reduced. We again have a flat reaction norm, and it's just the non-plastic genes that affect the phenotype. So we examined plasticity evolution under 4 scenarios. I won't go into the details of the equations, but I've left them up there in case people want to ask questions later. In the first scenario, we just have developmental limits. In the second scenario, there are additional costs associated with plasticity expression. In the third scenario, there is developmental noise associated with the plasticity component of the trait. And in the last scenario, there is environmental variation in addition to developmental limits. And today I'll just talk about scenarios 1 and 3. Qualitatively, scenarios 2 and 3 are quite similar, and scenarios 3 and 4 2 and 3 are quite similar, and 1 and 4 are quite similar qualitatively. So we present the results of evolutionary rescue in terms of how many populations survived at the end of 500 generations. And the value of the developmental limit on the x-axis here is scaled so that the size of the environmental change, if the developmental limit is equal to the size of the environmental change, then it has a value of 1. And in our simulations, evolutionary rescue or extinction happens within the first 50 generations. And for the rescued populations, the trait optimum is also achieved in that time. So for developmental limits only, we varied the magnitude of the plasticity expression parameter, B, which affects how much the plasticity alleles affect the steepness of the sigmoid. And we also varied the developmental limit. And what we see is for very small values of the developmental limit, it's virtually indistinguishable from a fixed phenotype, which we show as the solid line in both panels. And when the developmental limit gets closer to 1, the survival is nearly 100%, provided that the plasticity expression is high enough. So for the scenario with developmental noise, however, we varied the developmental noise parameter and set the plasticity parameter equal to 0.6, which makes the results comparable to the B equals 0.6 line on the left. And for the scenario with, and essentially as with previous work that didn't consider developmental limits, developmental noise didn't affect evolutionary rescue. And for genetic assimilation, we present the results in terms of relative plasticity of the rescued populations, which is essentially the contribution of the plastic trait component to the optimal phenotype. So these figures show how plasticity changes over time. All scenarios have an initial increase in plasticity that aids with rescue. However, for developmental limits only, increasing the size of the developmental limit increases relative plasticity, but it's not sufficient to induce genetic assimilation. And for very small values of the developmental limit, the ability for plasticity to evolve in the first place is constrained. So for developmental noise, the maximum plasticity and the subsequent reduction of plasticity are both related to the size of the developmental limit, as well as the noise parameter, which is similar to previous models with linear reaction norms. And for reasonable values of the noise parameter, genetic assimilation still takes hundreds of generations, which is at least an order of magnitude larger than what's been observed empirically. How am I doing on time?
5 minutes?
5 minutes? Oh, okay. Conclusions. For evolutionary rescue, developmental limits hinder evolutionary rescue. And as with previous models with linear reaction norms, plasticity costs hinder evolutionary rescue while developmental noise does not. And environmental variation doesn't affect evolutionary rescue except at very high levels, after which there's a very sharp drop-off in survival. And when it comes to adaptive plasticity, when the developmental limit is small, the evolution of adaptive plasticity is constrained in the first place. Therefore, evolution of non-plastic genes is necessary for the population to persist. And surprisingly, environmental variation doesn't affect plasticity evolution, even when it might favor plasticity. And we hypothesize that might be because of a mismatch between the linear environmental gradient and the nonlinear reaction norm. So when it comes to genetic assimilation, developmental limits aren't enough to induce genetic assimilation. And as with previous models with linear reaction norms, selection against plasticity is still necessary for genetic assimilation to happen, either in the form of plasticity costs or developmental noise. And environmental variation doesn't affect genetic assimilation. So our main question still remains unanswered, which is why doesn't genetic assimilation happen more quickly? So we offer several possible explanations in our manuscript. I'll just mention a few. So one possibility is maybe as we saw in the spadefoot toad example, maybe partial assimilation is the norm in natural populations, and full genetic assimilation only happens under certain circumstances. Another possibility is maybe the plastic response never goes away, but it's just not visible in the observable environmental range. So in the horn beetle example I showed earlier, genetic assimilation was observed through a shifting threshold, which in our model would be equivalent to shifting the position of the inflection point on the environmental axis. So in that case, you might have a situation where a population might appear to have a fixed phenotype at the original optimum, and then when it's subject to environmental change, the threshold shifts to the left and appears at first that there's a plastic response to the environment, but once the threshold is completely shifted to the other side, it again looks like a fixed phenotype in the novel environment. Another possibility is maybe it's the size of the developmental limit itself that is evolving, and genetic assimilation occurs through a reduction in the range of plastic trait variation. So these are also possible future directions for this model as well. So this work is currently in press in Evolution. If you're interested, I can send you a copy of the manuscript. I'd like to acknowledge the University of Florida Foundation that funded me during this work. And with that, I'll take any questions if you have any.
We have time for like one quick question.
Hi, very cool talk. I was curious if you looked at sort of accelerated and deaccelerated environmental changes at some point, just because that might be necessary for it to actually have some sort of, you're sort of quickly adapting, so initially you need that.
So you mean like a continuous change to the environment?
Yeah.
Yeah, we didn't look at that. So in all of our simulations, we look at a step change in the environment, an abrupt change.
Do you have any intuition for if that might be the reason?
I actually don't have any intuition for this, honestly. Do you have any?
No, it won't. In the other, well, with a linear reaction norm, we also looked at continuous variation. Doesn't make a difference.
Hello everyone, I'm Puneeth. I'm a PhD student with Matt Osmond at the University of Toronto. I'll be talking about one of my PhD projects, which looks at the role of epigenetics in evolutionary rescue. Before starting this talk, I want to acknowledge that This work was done at University of Toronto, which has traditionally been the land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit, and I'm grateful for the opportunity to work there. The talk focuses on evolutionary rescue, which we have already seen an introduction of that, and just a quick recap is if you have a population well adapted to the environment, and it's sort of the population is at its carrying capacity, a harsh change in the environment can lead to a decline in the population. However, there might be genetic mutations already present in the standing genetic variation or generated after environmental change that can potentially be better adapted to the new environment. And if these successfully spread, your population can survive the environmental change, and it's called evolutionary rescue. However, because these populations drop to low sizes before the spread, these can go extinct purely by chance, and hence we need models to understand what's the chance of survival. A lot of the models in evolutionary rescue have focused on sort of a pure genetic basis. There have been some models of plasticity, and we saw another one just previously. My interest in this sort of aspect of things was to mechanistically build up what causes these sort of potentially non-genetic components that can affect your survival chances, particularly focusing on epigenetics. So you can think of a genetic sequence that doesn't change. However, there might be there can be modifications to these genetics like methylations, histone modifications, phosphorylations that can potentially lead to phenotypic differences. These phenotypic differences, here is an example of a plant that shows sort of differences in leaf patterns, can react differently to the environmental changes. So here you can see differences because of pathogen exposure. And therefore, these epigenetic modifications can be very important, particularly relevant in the context of evolutionary rescue, because these modifications can be induced or removed by environmental cues, and therefore can form like the first step of adaptation to a first step of first step towards adapting to the new environment. So in order to model this, we started from a very simple population genetic model. You have your classic two alleles, two genetic alleles. Here the red big A allele is the wild type, and you have the green phenotype done by the little a allele, which will be the genetic mutant. So there's genetic mutations happening. But at the same time, there are the equivalent modified alleles for each of these two genetic alleles, which also can undergo genetic mutations. We allow for that to be potentially different. And then finally, the modification from the genetic to the epigenetic allele happens via a rate of we call IM. And then during vertical transmissions, a lot of these epigenetic modifications can be lost, and therefore we have a reset probability T as well. So one of the things which is well known is that these epigenetic modifications occur at a much higher probability than genetic mutations. And so we assume that M and T are of the order of 1 for some technical details. And then we assume that selection's weak and genetic mutations are also much weaker. So I'll just give a little bit of the model detail here just because it's sort of a lot of the work previously has been limited to sort of versions where only one of the genetic alleles can be modified because of sort of an analytical tractability. But sort of identifying that these M and T are happening at much faster timescales than the genetic mutations and selections allows us to simplify the model. So you can imagine that this little a and little b are happening at much faster, and so reach some sort of an equilibrium where they are present in the proportion of the rates at which they are being generated. And so you can think of these as a single effective allele, with their fitness being the weighted sum of the genetic and the epigenetic allele. Similarly, the same thing for big A, and this now reduces to sort of the classical PopGen two allele type model, and a lot of the analysis results from those can be exported. Further, we allow these epimutation rates and reset probabilities to be specific to the environment as well as to the allele, and we will see different scenarios with respect to that. Just to recollect, the red is the wild type phenotype, and therefore it's beneficial pre-change. So before the environmental change, the big A allele has the highest fitness, the little a has the lowest, and the epigenetically modified ones are in between. Post-environmental change, again, the mutant becomes the better one, and so more adapted to the new environment, and you have big A becoming the sort of the least fit. Okay. So coming to this sort of, usually when people talk about the idea that epigenetics can be beneficial in terms of these changing environment situations, what they assume is that usually the epigenetic modifications are only happening in sort of one direction, and that's in the direction of the beneficial allele. And so what I mean by this is in the first step, what we assumed is before the environmental change, you only have modifications of the mutant back to its epigenetically modified allele, and post-change, we only have sort of the wild type going into its epigenetically modified state. So all of the results will have sort of this same format, and so I'll just sort of quickly walk you through the axes. One thing which we found is that what mattered was not the exact values of m's and t's, but the ratio of m over m plus t. And one of the important points here is that if we plug t equals to 1, we enter the realm where there is no epigenetic inheritance across generations. And we see that this is not sort of a qualitatively different than having some amount of epigenetic inheritance, which I thought was interesting. We'll denote this ratio by m tilde. And what we will plot is the survival probability after this environmental change. And different values of the epigenetic modification pre-change is denoted by this color. If you had no epigenetics, the dashed line sort of sets the baseline in the absence of any epigenetics. And this is the result in this situation. And as expected, we see that all of the lines are above the dashed line, indicating that having epigenetics in this situation is always beneficial. Moreover, as we increase the epigenetic modification rate post-change of the wild type allele, we see sort of, again, an increase in survival probability. And so just a quick intuition, when we are increasing sort of post-change from capital A to capital B, we are increasing the fitness, effective fitness of the wild type, and that's the increase that we see. Similarly, as we go from blue to green, we are converting little a to little b before change, which increases its sort of presence in the standing genetic variation. And so that's sort of the basic intuition. This is sort of what people refer to when they talk of epigenetics being good for environmental changes. However, there's work that shows that epigenetic modifications can be linked to the genetic basis. So we look at the case where the big A has the same epigenetic modification rate pre and post change, and little a has the same epigenetic modification pre and post change. So everything else is the same, just that now there is sort of parameters that are equal to each other. And now we start to see some differences. So note that we have this region where your lines are actually going, some part of it is below the dotted line. So having an epigenetics in this scenario might not be always beneficial. However, increasing these epimutation of the wild type post-change is still increasing survival probability. So why do we see this decrease? It's because before the environmental change, while we do convert little a's to little b's, which here is a beneficial change, however, an unintended consequence of that is an increase in the conversion of little a's to little b's post the environmental change, and that's not good for the population because it's reducing the fitness of the genetic, effective fitness of the genetic mutant. And that's why we see sort of as we go here from blue to green, the rescue probabilities are actually decreasing. Okay. The other possibility is that what your environmental change is doing is it's changing the genome-wide epimodification rates. So here, what we assume is that the alleles have the same epimodification rates, just that they can differ between the environment. So in a given environment, there's the same genome-wide epimodification rates. Everything else in the model remains the same. And now things go haywire, and we see a bunch of different things happening. So now neither is that the epigenetics is always beneficial, so there is a large portion where it's not. But at the same time, somehow increasing the modification of the wild type after environmental change can potentially decrease these survival probabilities. So I'll just spend a minute over this. So what's happening is as we initially increase modifications of big A's to big B's, we're inadvertently increasing modifications of little a's to little b's, and that's what's causing the reduction initially, because you're reducing the fitness of the genetic mutant, the effective fitness of the genetic mutant. But when we further keep increasing big A's, what happens is we are converting big A's, enough of the big A's into big B's that they can compensate for it. So this portion of the part is where rescue is completely contingent on the epigenetic modifications, which is suppressing the genetic, which is not allowing the genetic modifications to occur in the population. So just to summarize these results, these epigenetic modifications might not always be beneficial for surviving environmental changes. This is because mechanistically, these modifications can either be linked because of the underlying genetics, or because these mechanisms are sort of altering the genome-wide modification rates, and these can have consequences for how these respond to environmental changes. The other thing which I think was worth emphasizing is that by identifying the separation of timescales between the epigenetic dynamics and the genetic dynamics, you can analyze a lot of these complex models by using results that already exist, which can be quite useful in terms of analyzing more stuff. So one of the things to do is, this is not specific for single allele haploid models. You can extend this to diploid as well as multi-locus models, and incorporate the effects of linkage and dominance and come up with these effective dominance and epistatic parameters. So I'm also interested in sort of extending this to sort of general non-genetic inheritance, including maternal and paternal effects and so on. And another point is sort of to extend this to a quantitative genetic type model and sort of mechanistically look at if epigenetics is the cause of plasticity, what kind of sort of reaction norms and stuff do you get, and see what sort of build up nonlinear forms of plasticity through the mechanistic aspects of it. I think with that, I want to thank my supervisor with whom this work was done, as well as the Osmond Lab, and happy to take any questions or feedback. Thank you. Yeah.
So you mentioned your model has stochastic extinction at
Yeah.
low population size.
Yeah.
But do you have genetic drift at any low population size affecting the surface selection?
So at the moment, the results, we pre-change, we're looking at a deterministic model, so there's no genetic drift. So post-change, it's sort of part of the demographic stochasticity. I just started running the stochastic pre-change stochastic parts. For those, most of the results seem to still hold. But of course, because these M and T are happening at sort of much rapid scales, any sort of minor changes in drifts can potentially sort of amplify up to that point. But at the moment, I haven't seen that happening, but that's one thing that I'm trying to make sure is not a huge difference. But yeah, this is good. Thanks.